I'm James Gagnon. I'm an assistant professor at the University of Utah. I'm here to tell you about nature's sunscreens. So I probably don't need to remind you, as people living in Utah, that the sun is great. The sun provides heat, the sun is powering photosynthesis on our planet, and because of these two facts, sunlit environments are filled with food. And so because of that, most organisms on Earth live in sunlit environments. However, you probably also know that the sun can be bad. And that primarily is because, in addition to producing visible wavelengths of light, the sun produces ultraviolet radiation that can penetrate our atmosphere and damage living systems. So UVB and A reach the surface of Earth, and they can cause DNA damage and oxidative stress to our cells that can lead to cell death and can cause skin cancer. And Utah has one of the highest rates of skin cancer in the nation, which fits with our beautiful 320 days of sun every year. Okay. So because of these challenging pressures from the sun, organisms on Earth have evolved a variety of protective mechanisms that defend them against ultraviolet radiation. So some organisms use nocturnal lifestyles to avoid living in the sun. Other organisms coat themselves with scales, feathers, or opaque other structures to protect themselves. Many living organisms have enzymes that can repair damage to DNA before it causes a mutation. But organisms can also develop their own sunscreens. So if you're like me, you put sunscreen on before you go outside on a sunny day. But many organisms can actually synthesize their own sunscreens, and that's what I want to tell you about today. So we've known that sunscreens are widespread in the living world. Organisms like these bacteria on the left can produce a sunscreen molecule called cytonnemin, which has this brown color but also absorbs wavelengths of the ultraviolet radiation. Plants produce molecules called synnipate esters that protect themselves from similar ultraviolet rays from our sun. In animals, we only know about one sunscreen, melanin, which provides pigmentation in human skin. And in humans, this pigmentation can actually protect us from the damage from ultraviolet radiation. Many organisms use melanin as a source of camouflage, creating stripes or other patterns. So on the right here, you're looking at a tiny zebrafish larva. This is the organism that I study in my research lab at the University of Utah. And it has melanophores that produce melanin that coat the surface of its body and also pigment its eyes, and that's important for its vision. And so this, again, is the only sunscreen that we know about in animals. But the interesting thing is there are lots of compounds that absorb ultraviolet radiation in animals. Are those also potentially sunscreens? So one of these compounds is called gagisol. It was first discovered about 40 years ago in the eggs of cods, the fish cod. That fish has a Latin name, Gadus morhua. And so somewhat unimaginatively, the scientists named this enol compound gagisol. I would have chosen a different name, but I'm stuck with it. So this is the molecular structure of gagisol. It has this carbon ring with one double bond in the middle. And if you ask an organic chemist what this molecule does, they'd say it would be great at absorbing ultraviolet radiation. And they would be right. So on the right is a diagram of the absorbance spectra of this molecule. And you can see its absorbance peaks around 294 nanometers. That's square in the middle of ultraviolet B rays. These are the most damaging rays that reach the surface of Earth. And you can also see no absorbance at these larger wavelengths of visible light. And that's because this molecule, that's the property of this molecule that makes it transparent. So it absorbs UVB, but it is transparent because it doesn't absorb in these visible spectra. Okay, so we know that fish eggs in particular are chock full of this molecule. So we had two questions when we started this project. First, where is this molecule coming from? And second, is it acting as a sunscreen? So the first insight we found was that there's these two genes that encode enzymes called EEVS and MTOX. And they convert this linear molecule that you can see on the left side of the slide. This is a byproduct of the pentose phosphate pathway found in all living organisms. And it first circularizes this molecule and then creates that double bond to produce Gaggisol. And the amazing insight from previous researchers was that many vertebrate species have the genes that encode these two enzymes, meaning they might be capable of producing this sunscreen on their own. And so we set about to test this hypothesis. And the way we did this is using the CRISPR system that you just heard about. And so we made a mutant animal, a mutant zebrafish, that is mutated for the first enzyme in this pathway, the EEVS enzyme. So surprisingly to us at first, these mutant animals that cannot synthesize the EEVS gene, they are totally happy in my fish facility. We have these large aquaria. Those animals, those mutant animals, grow up just fine. They grow up to adults, males and females. They're perfectly fertile. However, I should remind you that my fish facility is not the natural world. My fish facility has LED lights that do not produce UVB. It only produces visible light. So these fish have not been challenged with UVB. We're going to do that experiment in a second, but first I want to show you that these fish lack Gaggisol. So the way we did this experiment requires me to introduce a little trick of genetics. So as you may know, all the compounds in an egg are actually deposited by mother into that egg during the process of egg production. So these RNAs, proteins, and small molecules are all deposited by mom into the egg, and they run the show of embryo development until the genome can be activated and start to produce new gene products. And so we use this trick to produce eggs that are the same genotype, meaning the genetics are identical, but they either contain maternally provided Gaggisol or they do not. So the cross at the top between a wild-type zebrafish and a male zebrafish that lacks the EEVS genes creates eggs that are heterozygous for the EEVS mutation, and they contain Gaggisol. The cross on the bottom, where mom is homozygous mutant for EEVS, crossed to a male that has both copies of the gene, also produces embryos with the same genotype, but they should lack the maternally contributed EEVS and any Gaggisol that was produced by this gene. And that's exactly what we saw. So showing you a little bit of mass spec data here, the arrow points to the mass spec peak of the exact mass of the Gaggisol molecule, the atomic mass. And you can see from the first cross, we see lots of Gaggisol as well as some breakdown products to the left of that peak. On the bottom, you can see the arrow pointing to a non-existent peak, meaning that these eggs do not contain Gaggisol. And this also tells us that these fish are producing this small molecule through their own synthesis. They're not accumulating it from their diets because these fish are all fed the same diet. Okay, so now we know that this gene is required for the fish to synthesize this small molecule. Now, let's figure out if it's a sunscreen. Okay, so we built a very sophisticated device. You've heard a lot about sophisticated devices today, and I want to tell you about the one we built in my lab. We took this old UV transilluminator that was sitting in the corner of the lab because no one uses these anymore. We turned it upside down, and we built a small wooden box. And then we put our zebrafish embryos in a tiny petri dish under that. And we had a little UV meter so we could measure the dose of ultraviolet radiation. And the first thing we did is we took wild-type normal zebrafish embryos that contain Gaggisol, and we gave them increasing doses of UVB. And we found a dose where normal embryos that contain Gaggisol have a modest impact in survival. And that was our favorite dose where we were going to challenge embryos that do or don't contain this small molecule sunscreen. So I guess I should mention that this dose is a physiologically relevant amount of UVB that a fish outside on a sunny day might experience in 10 to 20 minutes of exposure. Okay, so let me show you the data by which we tested the role of Gaggisol as a sunscreen. So here you're seeing two bars. The light gray bar is wild-type embryo survival in the absence of UVB. And you can see they survive really well. Now, when we turn the bulb on, giving them 450 joules of UVB, we see a modest decrease in survival, measured here by swim bladder inflation, which is basically a holistic measure of their happiness as a little zebrafish larva. Okay, so if we make those heterozygous embryos that still contain that maternally provided sunscreen molecule, these fish are also quite robust to UVB. However, when we do that reciprocal cross where the embryos lack maternally provided Gaggisol, the embryos are completely non-viable. And so I swear there really is a bar here on the right. But none of these fish survived. None of them inflated their swim bladder. None of them have an intact heartbeat. None of them elongate their tail. Any metric of viability, these fish are suffering due to UVB. If we make a cross where both copies of EVS are missing, these fish are also very susceptible to UVB. However, I want you to note that the light gray bars stay really high. And so in the absence of UVB exposure, these fish are perfectly happy. So this is really strong evidence that the small molecule produced by this gene, by larvae, is a really important sunscreen for their survival. Now, I don't have the time to tell you all this other data, but we've shown through similar experiments that this small molecule Gaggisol acts as a sunscreen to protect fish embryos for the first week of their life. And if they don't have Gaggisol, they get a lot of DNA damage. They have a strong stress response to that damage. This leads to increased cell death and ultimately organismal death. Okay, so what about melanin? Previously, I told you that melanin is known to be a sunscreen in humans. What about fish? If we look at a picture of a larval zebrafish at 2 days old, you can already see that they have these black pigmented cells that are containing melanin. And by 5 days of age, they have quite a lot more melanin. So what we did is a more sophisticated cross where we could generate animals that don't have melanin, don't have Gaggisol, or don't have both of these putative sunscreens. And this is the cross. I'm not showing you this because I want you to go through it. I'm showing it because my student is really proud of this cross, and it was not particularly easy to pull off. So he insisted I keep it in the presentation. Now, let's get to the data. So again, we're able to generate zebrafish larvae that either have melanin, don't have melanin, have Gaggisol, don't have Gaggisol. And when we measure their survival, you can see that the two sets of bar plots on the left, fish that contain Gaggisol are quite resistant to ultraviolet radiation, even in the absence of melanin. However, when they lose Gaggisol, there's only a modest effect of protection for melanin. So we think here Gaggisol, surprisingly, is a much more important sunscreen for these fish to survive. Now, I told you that these genes required to synthesize Gaggisol are widespread in vertebrates. We see them encoded in the zebrafish genome, other fish genomes, and a variety of other genomes. They're not present in mammalian genomes, unfortunately. It would be great if we could avoid putting sunscreen on every day. But mammalian embryos develop in utero. So their embryos are actually protected by the opacity of the mother's body, so they don't need to produce this sunscreen. But we started to wonder if there are any fish species that can't produce Gaggisol. And so we did a survey of published fish genome sequences, and we discovered that when we look across a phylogenetic tree of fish, we discovered many independent losses of the genes to produce Gaggisol. And we saw a very strong correlation between fish species that live in the dark, fish species that, for example, live in caves, deep sea environments, or murky waters. These fish tended to have lost the genes required for Gaggisol synthesis. And so this suggests that this sunscreen is dispensable if you live in a dark environment. Okay, so today I told you about a new sunscreen called Gaggisol. And in fish, this is maternally provided into the egg so that it can protect the developing embryo from the dangers of the sun. It provides robust protection against DNA damage and cellular stress, just like you would expect a sunscreen to. And it's a more important sunscreen than melanin or the actual eggshell itself. And I also showed you a little bit of evolutionary data that suggests that Gaggisol has been repeatedly lost when fish move into dark environments. So we're really excited about this sunscreen and the potential applications of a novel natural sunscreen that might be useful for humans. And I'd love to talk to any of you who are interested in working on that with us. Now, before I end, I have to show you some pictures of the people who did this work. First, I want to highlight Marlon Rice. Marlon was born in Salem, Utah, was educated by my colleagues at Utah State, and then came to do his PhD at the University of Utah. And he led this project from start to finish and is a real tribute to the educational system we built for STEM students here in the state of Utah. And I also want to highlight Julaine Machado, a undergraduate at the University of Utah, also from Salt Lake City, who really helped us get this project done. And we had some help from a few other students as well. So thank you so much. I really appreciate your attention.